So I was up at 4 o'clock this morning, 4 a.m. Partly because of the talks I heard yesterday, and I had some ideas, of course, because you see talks and you figure, I have a slide that will talk about that particular issue. So there was some talk about dementia, there's talks about emergent systems, there's talks about complexity and so on. And I had slides for all those things that I didn't use. So I decided I'm going to revise my talk. So I spent the next 3 hours revising my slides, putting all these new slides in there and making a presentation that would take about 4.5 hours to get through. So I spent the next hour cutting back the slides, ended up with exactly the same presentation I had when I started the process. So it's good to be certain, right, that you actually know what you want to talk about and you're damn sure you're going to talk about that particular thing. So here we go. My presentation hopefully will be done in 45 minutes. There might be some stuff I'll have to skip, but we'll see how it goes. How many of you went to the workshop in Austria in November on multimodal or multi-scale brain? One person, that's it? Okay, so you can be quiet because you've heard most of this probably already from Anna Solodkin. But anyways, I'm going to try and give you sort of an overview of our platform that we've been developing. And you'll hear a lot of themes that you've heard previously in the last this morning, as well as yesterday morning's presentation, particularly to do with modeling and how we think about how to use modeling to understand brain function. So we'll talk first about theories and architecture for our particular platform in the context of computational modeling in neuroscience. And then go through applications. First of all, just to look at the intrinsic activity of the brain. Those of you who are familiar with functional MRI literature, there's a lot of work being done on resting state activity right now. So we'll talk a bit about that and how we can use modeling to understand those things. Also to be able to merge multimodal data sets, particularly in this particular case, EEG and fMRI data. And then an application to stroke prognosis, and then finish off with an application to tracking epilepsy seizure propagation. Everything I'm going to be talking about today, yes, has been published, so you'll see some notes in the slides. I think the slides will probably be somewhere at some point, so you can also get the references from there, or you can ask me afterwards, and I'm happy to send you preprints as well. So Mark yesterday mentioned about the purposes for modeling. I'm just going to reiterate some of those points to you today. As a psychologist, which is what my training is, very often people will propose models, and especially in cognitive psychology is very bad at this. They propose models based only on the words. So for example, you make a model of memory function, for example, and it's sort of expressing what you think might happen, but there's not really anything tangible in that model. So to convert that expression of your thoughts to a model actually makes it much more tangible and testable, and very apparent to other colleagues. So you can then read that paper, take that model, adapt it for your own research, and then grow that model and test it in your own data as well. So it's a very explicit expression of what your intuition is or interpretation of particular data sets. We heard this morning, and we'll talk about it a bit later on as well, that modeling allows you to merge different pieces of data. And certainly the HBP is an epitome of that in the sense of merging data from multiple cells, multiple preparations, and trying to build a large-scale model from different pieces of data. So it really allows you to pull different pieces of data and develop a recipe, if you will, for how the system you're modeling actually works. In our particular case, the brain. And then finally, it helps constrain possibilities. Modeling allows you to sort of focus on what's essential to understand an issue, perhaps also to know what's not essential to understand an issue. Helps you focus your question on what really is important to understand what's happening. And there's a validation cycle that's inherent in that. You go back and forth between your development of the model, the actualization of the model to see how it's working, and then refining it later on once it's been applied in the empirical world. Modeling is good. The people who designed the airplane wings that I used, at least to get over here, or at least people in the plane used, of course, went through a lot of simulations to make sure that the wings were going to actually do what they were supposed to do. And modeling actually pushed the aeronautics industry light years ahead. So it helps both in terms of developing new models, but also to make sure that they're safe, which is good. When you're playing, you want to make sure you're safe. So it's quite useful both in terms of, again, testing theory, but also practically speaking in terms of being able to use it in terms of engineering and actualization of particular things. Now, we have an additional challenge in studying the brain is that we're modeling a very complex system. And complex system here, I'm using it in very, the definition of complex adaptive systems, for example. So it's actually very specific to that definition. I'm sorry. So here is a slide that I borrowed from a colleague at the New England Complex Systems Institute that talks about the general features of complex systems. So for example, and you'll hear some of these things mentioned later on in my talk as well. So you've got scales over which the system is organized. There are simple systems that interact with one another. The idea of networks is pervasive across all these different levels. You've got notions of emergent features of systems, and the level of interactions that's happening across these different scales. And those interactions are what is important to actualize the system. Multiple spatial scales, multiple temporal scales influence one another. And the modeling actually forces you to bring an appreciation for those particular aspects of how the system's organized. Spatial scales, temporal scales, emergence, and so on. And not surprisingly, the brain is in fact a very good example of a complex adaptive system. It's actually architecture specifically to be an adaptive system, you could argue at least. So how do we model that? There are dominant strategies. We've heard some about this already. I'm not going to go into too much detail. There's the top-down approach, which you heard about this morning. Artificial intelligence is a good example of that. PDP models. There's a colleague of mine at Waterloo, Chris Elias-Smith, who's been working on a project called NINGO, or SPAWN, which is in fact a very similar kind of top-down model. There's more a bottom-up approach. In some respects, the Human Brain Project course is in fact that, building the brain from the individual cells all the way up to the systems. And virtual brain, which I'll talk about in a second, is an example of that as well. One of the challenges in modeling complex adaptive systems, or complex systems in general, is really captured very well in a paper by Eeg and Goyard in 2006 in Nature Reviews Neuroscience. And this diagram just really well illustrates the indeterminacy that happens when you're translating between different scales in the system. I'm not going to go into too much detail, but basically you have manifolds that describe a surface, and then the parameters that express that particular manifold, and the transitions between different spaces in the manifold as well. The feature, not a bug, a feature of this particular space is there are a number of parameters that get you into that particular part of the manifold. So it's not a very good specificity to be able to know where you are in this space. Multiple parameters can give you exactly the same higher level behavior. So it's an indeterminacy between understanding how things at one level scale to things at another level. And that's a problem for modeling. It's a problem for trying to build a model from the bottom up. So to do that, you have to make an approximation of what you're going to ignore in terms of the features of the model to allow you to at least make some speculations, if you will, of the behavior in this space. So the last part here is really the key point I want to make about this particular slide. That for modeling, you need to decide on the critical features you want to explore, and importantly, which ones you're going to ignore. And make that very explicit when you're talking about your models to other people. I hear music. This is the platform here. It's open source. You can download it right now if you want. It's available for the Windows, Mac, Linux. You can download it directly. You can also get it through GitHub. It runs either as a GUI or it runs also through Python on a command line version as well. There is support online. You can't really see that, but there's a docs.virtualbrain.org that has all the documentation for the platform. This is the example here, how to download it. You can navigate through that. It gives you actual examples you can work through to understand how the software is put together. In addition, we do have workshops. There's one actually in a week and a half in Berlin We've had 6 thus far. There's another one happening at the INCF meeting in Montreal in the summer as well. And these are typically a combination of didactic and hands-on workshops as well. So if you want to learn about complex adaptive systems, want to learn about large-scale models, come to the Node workshop. It's actually quite fun. Okay, now, I mentioned the idea that in modeling behaviors in the brain, for example, you want to focus on a particular feature of the system. And in the case of VirtualBrain, what we're really working on is the philosophy, if you will, or the assumption that a lot of the mental operations that we're interested in looking at emerge from network operations. In our particular case, large-scale network operations. So the functions that we're looking at, cognition, for example, actually come from the distributed interactions between different parts of the brain. And each individual region is embedded into this architecture, so that therefore what happens in that particular region and its influence really depends on the rest of the system and the status of the rest of the system at that particular point in time, i.e. context matters. The important thing for the applicability of VirtualBrain is understanding that any perturbation of the system, whether it be damage from stroke, damage from head injury, or damage from disease, is going to have a distributed effect as well. So understand brain damage and disease, it would be worthwhile to have that network perspective, looking at how the networks change in the face of that insult. And that, as I said, is embedded in the platform itself. What we do, we have mean field approximations for populations of cells. There's a number of different kinds of versions of that. We have local connections, which are effectively instantaneous. Long-range connections, which have a time delay. We have a geometry that's, you can use a canonical brain or you can use an individual person's brain, and then connect it up in a wiring diagram, typically derived from diffusion tensor imaging or from anatomical literature as well. The workflow is something like this. You've got a cortical surface. That's the geometry essentially you're going to use to place the different nodes in your network, cortical and subcortical. The adjacency matrix or connectivity matrix, again derived either through diffusion tensor imaging, where you can estimate tractography between different regions. You can also use existing anatomical databases like COCOMAC, for example, to figure out what's connected to what. You introduce the dynamics through mean field approximations and generate you can use very simple phenomenological models, or you can use ones that are sort of spiking models and so on and so forth. You can stimulate nodes. I won't talk about that today, but you can actually stimulate focally into each individual node and see how it propagates, or multiple nodes at the same time. And then using forward solutions, you can generate the empirical activity you might be measuring from a system. For example, bull data, EEG data, local field potentials, as well as intracranial EEG data for epilepsy, for example. And you can see this actually can actually loop back, and you can update this process to try and improve the model's fit to the particular data set. There's a generic equation that captures the virtual brain. Again, the details depend on the particular mean field approximation you use. You can't read that on purpose. No, you can't read that. But let me just quickly go through it. You have a change in a population. This is the population at time t minus 1. You've got the local connections of adjacent populations of cells, then the long-range input from areas that are further apart. This is the stimulation term here, and this last term here is a noise term. It can be Gaussian, can be non-Gaussian, doesn't really matter. Additive multiplicative is also possible in the model. Shown in a sort of a graphic representation, you've got the long-range fibers here through diffusion tensor imaging, and you've got the local parameters here, excitatory and inhibitory populations of neurons, their mutual interactions, as well as the local connections between them as well. Again, the details depend somewhat on the mean field approximation you're using, but the representation is pretty much the same across different platforms, different local mean field approximations. One of the key things about the platform and how we're using it is introducing time delays in the model, so that the by and large, the distance of the connections are used to impose time delays between different parts of the brain. The time delays are quite important for getting data that represent or replicate empirical data quite well. So you'll see a pattern emerging, and the way the pattern transpires across time is really dependent upon the time delays, and that's incorporated into the model specifically. I'm just going to go through this very quickly. This is the GUI version with the cockpit, if you will, here, where you go through and specify the details of the model. Over here, you can look at the output of the simulation. This is the connectivity part of it. So here you're reading your adjacency matrix and look at how things are connected. I'm just going to blast through that. It's going to do a movie. You don't want to see that. Next one. This is representation of an Fmri bold on a rendered brain. You can change what the presentation looks like. You can save all these things as well if you want to use them for pretty slides later on. You can also dump it out and analyze them separately in your own package if you'd like. This is the EEG module generating this simulated EEG data. You've got the dural surface. You've got different locations of channels. And over here, you've got the EEG waveforms generated through a forward solution from the local field potentials. One thing that's important, and you'll see that in the next slide here. This is a movie that kind of goes through the process in a bit more detail. So this is a Vulcan. You can see the ears kind of pulled back. But you can do a triangulation of the cortical surface for an individual and specify the regions of interest, represent the different areas you're going to then model, and their foci. Again, using diffusion tensor, you can look at the connections and put the anatomical connections in there. In this particular case, you can also put the electrode cap on this person's head simulation, and then generate the data. So this is the simulated EEG data across this person's scalp. See a time scale there. And then in a second, you'll see the simulated bold data. The time scale, of course, is slower now. We're looking at hemodynamic response as opposed to electrical activity. So it's more sluggish. And then finally, the individual source activity as well. Again, the local field potentials. The important point here is that it's the same model that generates all these data. So it's quite a nice way to be able to integrate data sets because you can use the same mean field approximation to generate EEG, MEG, bold data, as well as in the case of epilepsy, surface or intracranial EEG as well. So it's a good platform to be able to integrate data sets, as I mentioned before. Okay, moving right along. Let me go through some examples now. How many people know about the whole idea of resting state networks in Fmri? Just 2? 12? Handful. Okay. So over the last decade, at least, if not longer, there's been a trend, if you will, to have people going to an Fmri scanner and do nothing, i.e. called resting state. And the notion there is that you can understand a lot of what's happening in terms of brain dynamics by looking at what happens during the resting state. That's an assumption. Whether it's true or not, we're still trying to figure that out. But there has been a lot of stuff published around resting state activity. And there has been, in this particular example, for example, characterizations of large-scale networks that are coherent during resting state and replicable across individuals as well. So you may have heard about default mode activity, you may have heard about attentional networks and so on. Those are typically derived through resting state analyses and then looked at in terms of population changes, in terms of clinical changes, relation to cognitive functions, and so on and so forth. For electrophysiologists who've been around for a while, you know that the brain is never really silent, so resting state is basically just intrinsic activity. It's always there, unless you're sleeping or really, really drunk. But it's always there, spontaneous activity, and it's a question, can you actually understand something about the function of the brain by looking at the intrinsic activity? The answer is yes, of course. There's been enough papers that suggest that's true. So what is it about the feature of resting state that's important to understand brain function? So we tried to model that over a number of different papers. There was a review article from Gustavo Deco, Victor Gersa, and myself in Nature Reviews Neuroscience, as well as in TINS, that talked about this whole field, as well as modeling the resting state activity. This, again, is an example of the workflow. And you've got DTI data here. You've got, again, the parcellation. You put these two together to generate the architecture for the model, generate local field potentials from the individual nodes, from their interactions, put it through a convolution to do a bold approximation, and generate simulated functional connectivity or the correlations of the time series that are generated from the simulations. And then fit them by modifying the different parameters, usually the connectivity strength, to fit the empirical functional connectivity. What we found very early on was that as you scale the parameters, in this particular case, the coupling strength, this is the fit between the empirical and the simulated functional connectivity. Fit here is measured as the Pearson correlation between the matrices. What you find is that the fit increases to a point, reaching some maximum, and then drops off quite dramatically as the coupling goes further along. So there's something about this area that's interesting in terms of what happens in the model there. What I'm going to tell you about, I'm not going to go into too much detail, but what happens in this subcritical point in the network is that at this point, the entropy of the system is highest. At this point, the propagation between different nodes over top of noise is also highest. This is basically an optimal working point in terms of the system architecture. And that's basically where it replicates the rest of the activity the best. So this characterization, the intrinsic activity, actually helps the brain get to a particular working point, allowing it to, in theory, process information maximally at a particular case in time. Now, most of the data that are out there that you'll see published talk about a very long time series for functional connectivity, like 20 minutes or so. And in 20 minutes, your brain does a lot of stuff. So is it possible then that the functional connectivity that you look at over 20 minutes is really not capturing the change in the network dynamics across that period of time? Now again, if you looked at EEG data, you know that there's a huge change in the patterns of interactions between channels or between sources over a very short period of time. And you can probably get the same information from Fmri if you do sort of a sliding window, looking at the dynamics of functional connectivity. And there's now, again, a whole small industry now trying to see if this dynamics of functional connectivity, first of all, is not artifact, and then trying to see if you can use that to understand the changing in the functional networks across time. So these are empirical data. Again, hard to see. The colored matrix, this is the actual structural connections. These are the functional connections across 20 minutes of scanning. Here you really can't see it, but across these windows, which is about 2 minutes, the functional connectivity pattern will change. If you look at each one of these windows and correlate them, you can create this sort of matrix here, which, if it was all the same, would be red everywhere. What you find is that along the diagonal, there is some correlations amongst adjacent patterns, but then it spreads out. So you have a changing functional connectivity or dynamics of functional connectivity across that 20-minute segment. Again, not terribly surprising, but nice to see. The important feature, which I'll just emphasize here, is that those changes actually reflect the exploration of a low-dimensional manifold of the system. And if you can characterize those clusters of functional networks as states, you can map the transition between certain configurations of functional networks between two parts, or at least two parts, of a broader manifold. So we have state alpha and state beta. State alpha has these networks. State beta has these networks configured. What we see happening across time spontaneously, again, is the system transitions between this configuration and that configuration. Again, spontaneously, just showing you that the brain is exploring, if you will, during this resting state activity. Now, we can do more with these kinds of experiments. There was the talk this morning mentioned the idea of trying to pull together MEG and Fmri, which is a difficult task, but has a lot of utility if it can be done. Fmri is a measure of hemodynamics. It's related to metabolism, essentially. So there are problems in terms of what you can ultimately measure from that. So you want to try and optimize your capacity to extract information by getting as much as you possibly can. So one way to do that is to do simultaneous EEG and fMRI studies, which are brutal to do, but are doable. If you have very dedicated students and compliant subjects, no problem. So this particular case, we took a data set that had both EEG and fMRI collected across 17 subjects. So we have DTI, we have the structural data, we have EEG, we have bold fMRI as well. We used the EEG data to drive the dynamics of the model, and then push that through a convolution again to fit it to the individual's bold data. So again, using their own EEG data to push through the model to fit back to their fMRI data. That just shows you the preparation. The notion here, again, in schematics is you do a source model of the EEG data to represent the EPSCs for a given cell population. And that drives the excitatory and the inhibitory populations in the local field model. You have the global input from the rest of the network driving these local oscillations. You then fit this back to the fMRI time series as well as the functional connections for all these regions as well. So again, the EEG drives the system, but you need to use that to fit back to the individual's fMRI data. Shown here again, local population, the source waveform schematically driving the local dynamics, and you have the rest of the network driving as well from a general input to the system. These are the fits to the fMRI time series. I know you can't see anything here, but you just trust me that in terms of fitting the fMRI time series, that the model shown in blue somewhere, no, sorry, black, fits better than the other variations of the model. We had some other control models to test with that random models, pure noise models, as well as using alpha power as a model, and the model itself was actually better than any of those. In terms of the functional connections, again, the model itself actually had a higher predictability of the empirical FC than the other control models as well. And you can see that here, where the correlation for the time series, the empirical in black here was higher than the rest of the models, including alpha power, and the functional connectivity was higher for the model itself than the rest of it. There are things to be learned from this model in terms of how these bars figure in terms of what's actually being fit to this model. And I can't go through that right now. The paper was just published in eLife about a month ago, so if you want to look it up, you can see what else happened. This is a really, really complicated paper, but it's worthwhile looking at because it gives you an idea of what you can do with this kind of modeling. So you can, for example, in this particular model, look at the alpha phase-based inhibition, power-based inhibition locally, explain the inverse relationship between the fMRI and resting state, or alpha, I should say. It does look at the link between these features and the local excitation inhibition balance. And there also, as a bonus, we got power law scaling emerging that's strictly related to the network dynamics. If you remove the network dynamics, you don't get power law scaling at all. The model doesn't work on other things. For example, this frequency distribution, the spectrum looks kind of crappy, but beyond that, it does pretty good for some other aspects of the model. So again, it allows us to look at the integration between different data sets, in this particular case, EEG and fMRI acquired from the same person, and use that to understand the local dynamics of the system and how that may help emerge the intrinsic activity that we measure in resting state fMRI, for instance. So take-home message from that collection of studies, which covers about 8 or 10 papers. The idea from that is actually that the intrinsic activity or the resting state activity that you see in the brain is actually your brain exploring possibilities. You can use it, think about as predictive modeling, for example, which is in line with some thinking about free energy, for instance, is actually thinking about what can happen. This is happening all the time. The brain is predicting what's actually going to happen and trying to adjust itself to actually optimize that prediction going forward. Other interpretations are possible. That's most consistent with what we see right now with the model. We can talk about that later if you'd like. Okay. Let's talk about some applications in the clinical domain. We've used VirtualBrain thus far for stroke, for epilepsy, and we're just finishing up some work on dementia, both Parkinson's dementia as well as Alzheimer's disease. All of these have slightly different features, which you might expect since they're different diseases. But the nice thing about this is that they could all be appreciated from the network perspective in terms of what happens. So even though, for example, stroke is typically focal, the effects of the stroke are distributed, not surprisingly. Seizure foci in epilepsy, for most cases, are focal, but again, the impact of the seizure is going to be quite distributed. And you can capture aspects of the epilepsy by looking at network dynamics. For stroke, for example, let me get back to the specifics. This brain has had a stroke. This is a 55-year-old male who's had a stroke in the left hemisphere. You can see, hopefully, that this shouldn't be white, which is bad. So that's a stroke. So we can take these brains and put them into the VirtualBrain pipeline. Again, the nice thing about the platform is you can take individual data and push it through, making a model of an individual's brain, which is the power of the platform. So this is a bit blurry, actually, but again, you can take the empirical data as input, the DTI data from the individual, their structural MRI to get the T1 gray matter, white matter segmentation, for instance. If there's a severe lesion, we do what's called a VirtualBrain transplant to put in the tissue that's missing just to help with the registration of the data into a canonical space, and then remove that node once it's in that canonical space. Push these data through mean-field approximations. This is the stepness heuristic 3D model we're using here. And then generate local field potentials, and then again going back to looking at the individual bold data for the fitting function. Getting back to the representation of the VirtualBrain equation, in this particular application, we looked at both the balance of excitation and inhibition for the local fields, as well as the impact, again, of the network, the global coupling parameters, and how that changed across individuals. This was done both in terms of fitting, but also just looking at the parameter space for these different variables. So for example, if we look at the global parameters having to do with conduction speed here and coupling here, and look at the global variance of the system. So the global variance of the system can be used as a proxy for getting to a critical point. Because as a system approaches a critical point, its variance will go up somewhat. It's not a perfect prediction, but it's a good proxy for that behavior. So the heat map shows the total variance of the network. And you see as we scale the parameters here, velocity doesn't make much difference, but coupling does. This is where the point in the system where the coupling parameters generate the maximum variance in that particular system, with other parameters being held constant. That was a control. That's this person here. This is another control. You see the region where the variance gets high is similar to, although not identical to, the other control individual. This is an example from 3 patients who have had stroke. 2 of them show somewhat similar in a sense. It's shifted, but the general pattern is the same. Here, you see the morphology is different. Here, it's quite a bit different from the individuals. There were 35 stroke subjects and equal matched number of controls in terms of age. This is a heat map for the local coupling, for the NMDA coupling and GABA coupling. Again, parameter search, and you get this area of the search space where the variance, again, is highest, using that as the parameter estimate that you want to look at. You do that across individuals. Here's our 2 controls again. Again, the maximum point for the 2 controls is roughly in the same area. For the stroke subjects, you see that they're quite different. Again, not surprising because they do have a hole in their brain, but it's nice to see that there are some differences between them. The parameters are actually meaningful clinically in this particular case, and that's what's shown in the scatter plot below. The scatter plot below is a measure of the response to motor therapy for the stroke subjects. And this is immediately after therapy, and this is 1 year after therapy. And you see that the coupling parameters, and in this particular case, correlate negatively with the clinical outcomes. So those who have less excitatory coupling actually show better recovery of function immediately as well as later on. And that gets us to the idea of being able to personalize these models and have them have some clinical utility. You can use this, for example, as an estimation for prognosis for recovery of function. We are replicating this now across a much larger sample, of course, because this is only 40 subjects, not very big, but it is promising that we can use these parameters directly to, in a clinical sense. Okay, so the stroke model suggests that there's a change in the balance of local and global dynamics, and this balance predicts recovery. I forgot to mention that the G parameter of the global coupling also predicts recovery. It's not quite as strong as the local parameters. So there is both local and distributed aspects that are predictive of clinical outcome in this particular case. Also, if you look at just clinical scores themselves, they do not predict the recovery as well as the virtual brain model does. So there is some specificity as well that comes from these data. The last example is both a case study, but a case study that led to a large-scale clinical trial that's ongoing now in France. Here, the mandate is to use the network model to identify which nodes in the network are the ones that are likely generating the seizure and which nodes in the network are the one that propagates the seizure. And that's clinically relevant because if you're going to go in there and remove a seizure focus, you want to remove the one that's generating the seizure and not the propagation zone. So you want to minimize, obviously, the tissue that you removed so you have better recovery for the individual. So that's the goal. The key processes for virtualization, going back to what we talked about earlier, was to first of all get a topology for the connectivity, a topography for the regions of interest that are generated here, and then put in dynamics for the local areas. Dynamics here are generated with what is called the epileptor model. It's a phenomenological model, and it's derived specifically to characterize seizure activity in a given node. I'll show you the behavior of that in a moment. You can use a heat map to identify the epileptic zone, and then identify from overlaying, for example, the MRI itself, where the abnormality might actually be generated from. So this is the epileptic network, and this is a propagation pattern here. So here there's no reason to remove these nodes, whereas these nodes here would be candidates for excision later on. So these are the data that go into the model for an individual patient. This you've seen before. These are basically just their connectivity, their structural information getting pulled together into a network model. In addition, you want some estimation of activity. Now, it's a bit more difficult in epilepsy. You can do clinical EEG to get an overall measure of the scalp topography of the seizure. But what most often happens is they do some either surface EEG, or they take off the scalp and put a cortical sheet on top of the dura, or in France and in Cleveland, they put in surface needle electrodes. As you see here, these are needles that actually inserted into the brain, and they have multiple contacts that allow you to measure activity from a bunch of different cell populations along the needle. And from there, you can measure these from these needle electrodes over several days, sometimes even weeks, depending on how long the person is there. Hopefully, you catch a seizure and be able to look at then the propagation of the seizure to have an idea of where the seizure is coming from. So these are seizures that had symptomatic. They actually generated full seizing activity, where you have some seizures that happen, but they don't have sort of the full expression of the seizure, behavioral manifestation of the seizure. So this is some data from that particular patient, virtualized. This is her brain with the electrodes here. Each of the balls corresponds to one of the channels from the needle electrode. You'll see that the size of the ball corresponds to how much activity there is at a particular point in time. So you can kind of see, this light's changing as well. You can kind of see this slice here corresponds to a high activity in this blue channel, which is very close to the seizure focus, and high activity in some of the blue channels, which is the contralateral area here. It's kind of like strobing a little bit, isn't it? It's like, which is, I was going to say something about seizures and strobing, but I'm going to pass on that. So this is a complex partial seizure. You see the seizure onset here, and then propagating across to the purple channels, blue channels, and red channels, which are in the contralateral hemisphere. So they have multiple needles here. They have no needles on the other side. So we can simulate that. The task in the first model was to figure out how the seizure is actually propagating to the contralateral hemisphere, because it wasn't clear from the clinical data how the seizure got over to the other side. So seizure starts in the right hippocampus, and then as time goes forward, it spreads into thalamus. Now, in the data from the clinic, there was no needle in thalamus. But in the model itself, the virtual brain model, since it can measure from all the nodes, it said the best way to propagate that seizure is to actually have it go through thalamus. It then crosses over to the left hippocampus, temporal lobe, and then terminates. And we can put that into a movie. You really can't see very well again, but these things are changing as the activity propagates. So you can see over here, seizure onset in the right hippocampus, crossing over, propagating into the right hippocampus. And then to the temporal lobe. We should have some music to this one, shouldn't we? At least because you can't see it. You're going, what's going on there? I don't see anything. You need some music to kind of. Anyway. So the patient was brought back, and she was taken into a scanner again to do a more high-resolution structural scan. And they found that she had a hamartoma in her hypothalamus, which had strong connectivity between hippocampus, contralateral hippocampi, which is probably how the seizure was propagating. So it was actually an abnormal connectivity that she showed. And so in the follow-up, they included that additional connection in her data set and got a much better fit to the data. And now she's being followed up now for intervention, surgical intervention. So this whole process now, in terms of how it's being used in Marseille, now has the entire pipeline that they're testing for clinical utility. So they're now mobilizing a national clinical trial to use the virtual brain in concert with other interventions to guide how one might treat a patient. So they have been doing a lot of imaging, both in humans as well as animals. There was some mention yesterday, I think, about the Allen Mouse Brain Atlas. We have a version now in virtual brain as well, so there's a virtual mouse as well as virtual man in the software package. You can construct the avatar for an individual. There's standard pipelines that do that quite well. You introduce then the functional, the model for the regions, which is again the epileptor model, for example. And then refine the pathology based on fitting of the individual's data back to the particular model, done through using Bayesian optimization to identify the parameters that are most likely for a given set of parameters. You then have a chart that characterizes the most likely propagation zone as well as the epileptogenic zone for the individual, and that then drives decisions around how to intervene for that particular patient. So the clinicians are quite excited about this because they do like to have additional information to guide how they're going to intervene. And what they've seen thus far has been quite exciting because they do, in fact, it does help to constrain what they're going to cut, which helps with prognosis for the person going out. So the nice thing again about doing the virtual brain and doing the virtualization of an individual is you can intervene in the model first to see what interventions like the best outcome, and then use that again to constrain how are you going to treat the patient. So I've covered a lot of ground in a short period of time. I showed you the platform. There are workshops, as I mentioned, that you can come to if you like, or you can actually just download the thing yourself and figure it out, because it's not that difficult compared to other platforms that are out there, but it is a bit of a slog for some of us. You can use it to look at empirical data, humans as well as hopefully at some point mice and men. So as I mentioned, we do have a TVB mouse already. The macaque version is almost finished. We'll probably have that done by the summer. We are now putting in the multi-scale aspect of it to look at between EEG and Fmri. You can envision that actually now moving between different kinds of scales as well, all the way down to individual electrodes if you want in the mouse and in the macaque, for instance. The models I showed thus far have been looking at intrinsic activity, so there's no stimulation, i.e. looking at pictures, for example, or movies or hearing sounds. So cognitive stimulation is one of the things we're looking at now as well. There's a group in Washington, D.C. that's doing this already with working memory studies and doing it quite well. So there's some extensions there. And of course, the clinical applications. Epilepsy I mentioned already. It can also be used, the platform, for testing stimulation efficacy. So again, you have individual model, particularly white matter fiber tracts, which constrain the propagation of stimulation. You can include that in the model to look at stimulation propagation in an individual. And I mentioned dementia as one of our projects going on right now as well. This is an open source project. There is already linkages between Virtual Brain and Human Brain Project. It's evolving over as we speak right now, as a matter of fact. And it is a nice way to link theory and data, which is in fact the goal of modeling, and that's really why we're here today in a lot of cases. The other reason we're here today, this has to do with the title of the workshop, focusing on transdisciplinary neuroscience and the fact that neuroscience, as it's evolved over the last 30 or 40 years, has become the embodiment of working across disciplines. The reason I mention that is because this platform would not have been possible if it was one discipline only working. So for example, here are the key scientists that were involved in the platform. Since I'm giving the talk, I get to be at the first. So this is me. I'm a psychologist by training. Michael Breakspear in Australia, he's a psychiatrist, also a mathematician. Gustavo's a psychologist. Deco, he's also part of HBP and a mathematician. Carl, probably some of you might know, Friston, is a psychiatrist as well and an engineer. Victor's a physicist. Nancy is an MRI physicist. Mary Pat's a neuropsychologist, clinical training. Kathy Price, also a neuropsychologist. Petra Ritter's a neurologist. Steve Small's a neurologist and is an anatomist. And Olaf actually was a molecular biologist when he first started. And now he's a network dude. But you get the idea that the success for platforms like this really require the confluence of different disciplines. And that's really what I think spoke to our success. We've been going now since about 2006, and actually will be important for the success of HBP going forward as well. So great job to pull this idea together. This is exactly what you need to ensure the success of HBP going forward. So I'll end there. These are points you can read while I'm drinking water, and I'll take any questions you might have. So thank you for your attention.